Hi, Miss Nikki here. This is our Chapter 4 kind of review. Let's go into cellular level of organization and talk specifically about cell theory and why cells are small. So you should understand the three basic tenets of cell theory. A cell is a basic unit of life. All organisms are made up of cells. New cells arise only from pre-existing cells. And we'll talk more about mitosis, or cells copying themselves, when we get to Unit 3. Also, there's a little section about microscopes and looking at plant and animal and actually being able to take the leaf and look at the cells underneath the microscope. We do have a microscope lab, virtual lab, that you'll be completing, so you should be able to get a good sense of what it looks like underneath a microscope when you place a sample. What I want to talk about in a little more detail is this surface to area, or surface area to volume ratio. So when they say surface area, they mean the outer surface of an item. So probably the best way to explain that is all of this on the outside, this is surface area. Everything inside is volume. So this is, say this is a large cell, right? So this is one really, really large cell. And these are smaller, and then there's there's one back there you can't see, right? So now this is the surface area, and inside is the volume. So what they want you to understand is that cells have to stay small because cells need surface area and large enough to allow sufficient nutrients to enter and exit. So what does that mumbo jumbo mean? Remember, we have our cells, and cells have a plasma membrane. And the plasma membrane is the area or the fence where items can enter and exit the cell. So this is your barrier that either allows items inside the cell or out. And remember, what are cells doing? They're constantly undergoing something called metabolism. So they're constantly breaking down or putting together molecules. Again, remember dehydration, synthesis, and hydrolysis. So if I try to make this a little bit more clear, this is, let's say, an apple. If I were to skin this apple, I would have a surface area about this length. Say these are a bunch of grapes, and we might have much more than just these 8 cubes. But if I were to take the surface area off of those, I would actually have more surface area per volume. And that's what they're trying to show you here. Surface area per volume. So I have more surface area length compared to the volume that's inside. So having a larger surface area to volume ratio allows for nutrients and wastes to enter and exit a plasma membrane of a cell. Okay, we talked a little bit about this in a previous lecture, the difference between a eukaryotic cell and a prokaryotic cell. So a eukaryotic cell has a nucleus, so eukarya meaning nucleus, pro meaning before. And remember, we already talked about bacteria and archaea. We also talked about protista and fungi, and then plant and animal. Those were the kingdoms. Good. All right, so a eukaryotic cell has a nucleus and a prokaryotic cell does not have a nucleus. They both have DNA, though. Let's just make sure we clarify. They both have DNA. I guess I can put this in slideshow, and then I would be able to move from slide to slide, hopefully. No. Okay, so this is trying to show you a difference between a eukaryotic and a prokaryotic cell, and also talk about size differences. So you can see in this picture that the eukaryotic cell is much larger than the prokaryotic cell. And here is the nucleus. We can see the nucleus within the eukaryotic cell. The DNA would be free-floating inside this prokaryotic cell, and we'll see that in just a second. So some differences about prokaryotic structure. There's no membrane-bound nucleus. There is a plasma membrane. There are ribosomes which make proteins. They have DNA. They might have a cell wall, and they're going to have attachments. These are like hairs, pili, and flagella. So here's a picture of kind of your typical prokaryotic cell. You can see here is the DNA, and the DNA is just free floating. It's not encased inside of a nucleus. You have ribosomes. These are going to help make proteins. Remember, we went through the DNA is going to copy itself into RNA, and then that RNA is going to make proteins. Well, you need ribosomes right here to turn that RNA into a protein. We'll talk about that a little bit later. There's always a plasma membrane. That's this light blue structure. There's a cell wall, which helps give the prokaryotic rigidity or strength. And then usually it has this capsule, and sometimes this capsule can be sticky. Again, if you're a prokaryote and you need nutrients or you need to survive off of a host, you have to have a way to stick to it, right? So there's usually some sort of sticky glycoprotein made, and there's usually these little hairs with which we can attach. And then there's some sort of way of locomotion or movement, and we call that a flagellum. Not all prokaryotes have a flagellum, though. This is just kind of the standard basic. So what about eukarya, the domain eukarya? So underneath the domain eukarya, we have 4 kingdoms, which is protista, plant, fungi, and animalia, or animals. What's different about eukaryotes is that everybody has a compartment, and those compartments are called organelles. So there's compartments within the cell, and those organelles have specific jobs. So you can see this is an animal cell, and this is much more complex. We have a lot of little areas that are carrying out specific functions. One of the kind of analogies is, think of a prokaryotic cell as just a warehouse. It's just a giant room. There's no sectioning off. Think of an animal cell or a plant cell or a eukaryote as an office building with many, many offices inside of it, where each office, maybe one has to do with billing and one has to do with payroll. So that's how these organelles behave. They have specific jobs. So I want to go through some of these organelles. So the first thing we have here, this is a nucleus. This is an animal cell. Right away, you can always tell if something is prokaryote or eukaryote. Look for a nucleus. If there's a nucleus, it has to be eukaryote. If it doesn't have a nucleus, it has to be prokaryote. So this right here is called the nucleolus, and the nucleolus helps make the ribosomes. Remember, the ribosomes are what's going to help turn RNA into a protein. There's kind of this stringy substance. I'm not sure if you can see it really well on this picture, but this stringy substance is called chromatin. Chromatin is the same thing as DNA. Usually when we see DNA, you might see it as chromosomes later. Chromosomes. Let me write it. It's hard to write it like this. Or you might see it stringy, and this is called chromatin. It's all the same thing. You also have something called endoplasmic reticulum, and there's rough and smooth. So smooth has to do with making lipids, and rough has to do with making proteins. It's studded with ribosomes. So you can see here there's little ribosomes attached to this sac-like structure, the endoplasmic reticulum, and that makes proteins. And then the smooth endoplasmic reticulum, which is here, helps with lipid production. We also have mitochondria. This is where we're going to make ATP. This is very important, coming up in one of our, I think, Chapter 7 lecture. You have lysosomes. Basically, lysosomes have digestive molecules in them. So digestive enzymes that can break apart items that need to be discarded. So if something inside the animal cell is not working properly, the lysosome can gobble it up, chew it up, spit it out as waste. This out here is called the Golgi apparatus. The Golgi apparatus is for packaging and processing and modifying proteins that this cell might make. We'll see a little bit in this in a second. Vesicles, basically this is how you transport something inside of a cell. Cytoplasm, this is kind of a fluid, a semi-fluid. It helps give support for the organelles. You have something called a centrosome. Centrosome will come up later, and centrioles. Both of these have to do with division. So if we're going to divide this cell, we have to have these centrosomes and centrioles. Cytoskeleton, this is like the scaffolding of the cell. We'll talk in just a little bit about some of these filaments and how they act as a supportive. So basically it's the backbone of the cell. It's the scaffolding. It's like the bones in our body. It kind of holds the cell in its shape. And then, of course, we have our plasma membrane that we've already talked about. We remember that this is a bilayer phospholipid, right? So here is a plant cell. One of the things I want you to notice are similarities and differences. So plasma membrane is in both. So plant and animal both have plasma membranes. Both have mitochondria. Both have cytoskeleton. Golgi apparatus, endoplasmic reticulum, cytoplasm. The differences, and they have little stars here. So the differences, chloroplast and a cell wall. Both of these you find in plant only. So chloroplast is in plants only, and this is where we turn sunlight into glucose. So cell wall and chloroplast you find only in a plant cell. And then also you have this central vacuole. This is a large sac that can store substances, usually water. This central vacuole, if a plant is drooping, its central vacuole is low or not filled as much. If a plant is standing straight up, it's not wilted, it has plenty of water and its central vacuole is full. We can also see there's still a nucleus. There's still DNA inside this nucleus. So you should know the items that are different in a plant cell, and again they all have little stars next to them. So you can see chloroplast, cell wall, central vacuole. So let's talk a little bit about the nucleus and the endomembrane system. I just want you to see kind of the flow of information, how it starts the nucleus and works its way out. So here is the nuclear envelope. The nuclear envelope is around the nucleus, and it has these little pores, these nuclear pores that allow DNA, we're going to see in a second, which is going to copy itself to RNA, and that RNA is going to be threaded out of these little holes. Now remember, what's RNA going to become? RNA is going to get made into a protein by ribosomes. So this is the big picture here. Here's our nucleus, here's our DNA. Our DNA gets copied into RNA. Right now, don't worry about the M, it just means messenger RNA. The RNA gets threaded out of this nuclear pore. And what's going to happen, these are ribosomes. Remember I told you ribosomes take RNA and turn it into a protein. Then this protein gets dropped off into the endoplasmic reticulum, and this protein is going to be folded. Or processed, however you want to think about it. Remember when we talked about primary, secondary, tertiary structure, we talked about the fact that proteins are not fully functional until they're folded correctly. So this is where that folding takes place. The next part here, they're trying to show you just kind of a blown up version of the rough endoplasmic reticulum. So you can see the nuclear pores right here and here. So the messenger RNA would be threaded through this rough endoplasmic reticulum, and this is where the ribosomes would help turn that messenger RNA into a protein. Once we have a protein, we put it in a vesicle. So we went DNA here, we went messenger RNA here. Then with the help of ribosomes, we turn the messenger RNA into a protein. Now that protein gets shipped in a vesicle to the Golgi apparatus. Sometimes we need to make further modifications to a protein to make it functional, and that's where it happens. That's the Golgi apparatus. Once the protein is fully functional, we're going to put it in a vesicle and send it somewhere inside the cell or somewhere outside of the cell. So a cell can make proteins for itself, and it can also make proteins to excrete to the outside of a cell. So this is just a larger picture showing you incoming and outgoing. But what I'm kind of most concerned with here is the outgoing. So we're here at DNA. then we have messenger RNA, then we have a protein, and then we can have that protein be modified, get it out, and then we can have that protein shipped. And that protein can be shipped inside, outside, or inside the cell. All right, vacuoles and vesicles. I've already talked about vacuoles. I just want to make sure that you understand what the central vacuole is. It's this structure right here. As far as chloroplasts and mitochondria, these two are very, very important in energy function. When we talk about chapter 6, we're going to talk about photosynthesis and all about chloroplasts. When we talk about chapter 7, we're going to talk all about mitochondria and cellular respiration. So basically, these structures are responsible for our energy needs. So go back to one of the first chapters. We said we have sunlight, and the plants take sunlight and they turn it into glucose, and then we pull the plant and eat the glucose, and when we eat the glucose, we take the glucose and we turn it into ATP, and this allows us to do work like muscle contraction. So we are interdependent on mitochondria and chloroplasts. I wanted to put in kind of this origin, this endosymbiotic origin of chloroplasts and mitochondria, because both of these structures have two outer layers, or I should say an inner layer and an outer layer. But because they are double membrane bound, very much like our nucleus has a double layered membrane, they think that these used to be prokaryotes, that they were independent, and that somehow they got absorbed into the evolving, I can't get it out, evolving eukaryote. Now again, this is just a theory, but this is currently what we do believe. They each of these have their own DNA. So you have DNA that's not inside your nucleus. It's either inside chloroplast if you're a plant, or mitochondria if you're an animal. So because they have a double layered membrane, and because they have their own DNA to help make the proteins necessary for photosynthesis and cellular respiration, we believe that they were prokaryotes at one time that were incorporated into a eukaryotic structure. Cytoskeleton and movement. Okay, so this is one example of how we're going to use this ATP to do muscle action. So this is called actin filament. We have this in muscles, along with something called myosin. And ATP has to bind in order for this head to reach up, grab this actin, and pull. So ATP allows for these molecules to pull on each other, and this generates muscle contraction. So usually if a muscle is long, it's relaxed. And when it pulls like this, it becomes shorter, and this is contraction. You have to have ATP in order for this action to occur. We also have other filaments, intermediate sized filaments and microtubules. Basically, I just wanted you to see that these filaments have to do with the microtubules and the proteins that are available inside the cell, either animal or plant cell. So these all fall under cytoskeleton, which these are going to help maintain shape, or they're going to somehow assist in movement. So probably one of the best examples of the movement part is the flagella. We also have hair-like structures, so this is a good example of hair-like structures. They kind of sweep out debris or any pathogens. You can find these cilia in the lining of your windpipe, or what we call your trachea. You have it in the lining of your bronchioles, the tubes that go to your lungs. And then this here is a sperm cell, and they're trying to show you the sperm cell with its flagella. So the flagella is just a very orchestrated, I don't know how to word it exactly, orchestrated cytoskeleton filament arrangement that allows for this whip-like movement to occur. All right, eukaryotic cells in review. Go back to this table that's in your book and look at the description and the function of each cell structure and make sure that you know a little bit about each of them. Well, I think that's it for Chapter 4. I'll talk to you guys in Chapter 5. Bye.